In the last part of the video, we will be talking about the locomotion of cells. The first we have the amoeboid movement and the second we have the cilia and the ciliary movement. The amoeboid movement gets its name from an organism itself called amoeba. First, the cell forms a pseudopodium like amoeba does. These pseudopodiums will release exocytotic vesicles. These exocytotic vesicles are full of protein receptors. These protein receptors first will get attached to the pseudopodium, and when they reach near to the surrounding tissue or the cell, they will now attach to the ligand of that tissue. After this, some of these receptor proteins do not get attached at the ligand, but they enter again back into the cell on the other side of the cell. So the opposite side of the cell will form endocytotic vesicles. So there is endocytosis happening, and these endocytotic vesicles, when these are exiting, these will again form the exocytotic vesicle. This will lead to the other side of the cell to come in place where the pseudopodium was formed. Hence, the cell moves in the destined position, or the cycle goes on until it has reached to its position. For example, the cells which perform this movement are WBCs when they are formed into macrophages. Then we have fibroblast, sessile cells. Then we will have embryonic cells also. When the zygote has formed, it will go from the fallopian tube to the uterus for the further process. And the last will be the cancer cell. They can lead to metastasis of the cancer cells. For example, we have sarcomas. These are controlled by a process called chemotaxis. In chemotaxis, there will be release of special chemical substance called chemotactic substance. When they go from low to high concentration, it will be termed as positive chemotaxis, and if they will go from high to low, it will termed as negative chemotaxis. On whichever side these chemotactic substances are more, the cell will form pseudopodium on that side itself. The next is the cilia and the ciliary movement. It is divided into two, that is motile and non-motile. First, we will discuss about the non-motile. As the name suggests, it cannot move, and these are present as a single cilium on the cell. Although they do not move, but they have a function as a sensory antenna. For example, in case of kidneys, they are present on the epithelial cells of the tubules. When there will be a flow, it will act as a flow sensor. If these flow sensors are defective, it can lead to polycystic kidney disease. And in the motile form, as it suggests, it can move. And these are present in two parts of the human body. First is the respiratory membrane. It helps to move mucus at the rate of 1 centimeter per minute towards the pharynx and clears the unusual particles which we have inhaled. And in the fallopian tube, it moves the ovum released from the ovary towards the uterus. The cilia has a structure. This is the ciliary stalk. The ciliary stalk is covered by a membrane. Because of the tip, we have the whip-like movement. The ciliary stalk has a size of 2 to 4 nanometer above the cell membrane. So above the cell membrane, the ciliary stalk will be present, and which will be of 2 to 4 nanometers. And they grow from the basal body. Basal body has a origin from its rootlet, and there is a presence of basal plate in between the cell membrane and the ciliary stalk. And when we will take a cross section of the cilia, we will see such structure, that is 9 plus 2. They will be connected by axonems, these white lines. And here is the 2, 9 plus 2. They also have inner and outer arms, which is dynein arm. These dynein arm provide the cilia with the ATP. so it is full of ATP. And for the movement of cilia, it requires magnesium and calcium concentration and ATP concentration as well, which is received by the dynein arms. So that's all in this video.